Thanks for tuning in to my talk today. I'll be talking about carryover effects of nestling diet on post-breeding rusty blackbirds. My co-authors on this project were Carol Foss and Robert Cooper. Just before I start, I'd like to remind people that there's a Rusty Blackbird workshop that's going to happen on October 7th and 9th from 12 o'clock to 3 p.m. on each day. And during that workshop, we're hoping to come up with a range-wide conservation plan for rusty blackbirds. So please register for the workshop on our website at rustyblackbird.org. All right, I'm going to introduce first a couple of things. So what I'm going to talk about today is based off of research that's been conducted since 2009 in New Hampshire. And we just published a paper on a telemetry study. And I'll be first introducing that research, and then the study that I did is on diet composition, and that kind of naturally flows off of the work that we did that was published in 2020. So rusty blackbird nest success has been found to be relatively high throughout the range. It's about 0.5 or so in Alaska and in New England. In our study in New Hampshire, we found that 60-day fledgling survival is relatively high at about 0.5. And contrary to what a lot of other people have found with fledglings, telemetry studies with fledglings, is that in the first week, the mortality was actually unusually low. We had only about 3% of our 18 fledglings died in that first week, and a lot of them lasted the whole length of the study. Just for comparison, other songbirds, they found about anywhere from 11 to 90% mortality. So in the same study that we conducted in New Hampshire, we put transmitters on males and females as well, and we found that our 60-day female survival was relatively high. We actually only had one female die, and that was before the end of the nesting period, so the breeding post-fledgling survival of females was 100%. The 60-day male survival was low comparatively to the females, 0.79. However, the confidence interval was pretty wide, and actual survival could be anywhere from 0.5 to 1. And that's compared to, there's not a whole lot of telemetry studies that are done on adults post-breeding, and the one study that we found with birds sort of similar in size was barn swallows, and both males and females in that study had a survival of about 0.92. So with that same study, we also looked at selection, and so we looked at nest site selection, and then hatchier selection, habitat selection, and then adult selection. And what we found was that none of the selection was influenced by, or that survival wasn't influenced by selected habitat features in New Hampshire. So what were those selected habitat features for nesting and post-breeding? Rusty blackbirds were selecting softwood saplings, wetlands, and low slope in a 30-meter buffer, and increasing or decreasing distances to streams. So we concluded in that paper that it might not be breeding ground habitat, at least, that cause a decline, just because these things that they're selecting for are readily available in New Hampshire. So the biggest effect that we had was that fledglings in New Hampshire were 30 times more likely to be present every 50 meters closer to streams. And then the next biggest effect size was that they were 3 times more likely with every 10% increase in wetland cover in a 30-meter buffer. And so in many studies that have been done on fledglings, what's been found, and we didn't find this, but in other studies we found that fledglings are particularly vulnerable to predation in the first week. We suspect that because rusty blackbirds nest in such dense habitat, you can see that here in the photo in the bottom left, that perhaps their first week survival is pretty high because they're concealed. However, foraging becomes a lot more important than concealment as time goes on for fledglings. They have to learn how to forage for themselves. And while concealment is still somewhat important, they really need to be able to get from the nest to get to foraging sites fairly quickly. In our study in New Hampshire, we found that nest sites were about 150 meters from streams, which would be a potential foraging area, and that nests were within 75 meters, or at least 53% of them were, and then 17% of nests were about 200 meters from wetlands. So they have to kind of cover quite a large distance. So you can imagine that fledglings that are able to fly longer distance or in better condition might be in better condition. And so what are they eating during breeding? Their diet is predominantly wetland invertebrate larvae and adults, including mayflies, caddisflies, dragonflies, fly and larva beetles, and anything that they might run across. They're rather opportunistic when it comes to food. So our objectives for the nestling diet composition study was to figure out if diet composition affected nestlings' ability to move to foraging sites faster in the first week after fledging. And we also wanted to determine if nestling diet composition is related to adult body condition, and specifically males, because we found that males maybe might have had a lower survival than females. And then we wanted to determine if diet composition influences nest condition in any way. So our study sites were in the Androscoggin Valley in New Hampshire. The red stars are the sites, and we had quite a few territories per site. And this was in northern New Hampshire. So for our methods, we had to capture birds to put radio transmitters on them, and then also to draw blood for the diet isotope study. We ended up putting transmitters on 22 hatchier birds, 20 males and 20 females from 2010 to 2012, and then we followed them after fledging, and so we actually had to go through the data and make sure we weren't including data that were still when the birds were nesting. So we included data after the birds fledged of both adults and nestlings from June to August. We tried to collect one location per bird per day. That didn't always happen. And we took blood from the hatchiers and the adults when the nestlings were about 6 to 10 days old. We generally took nestlings out of the nest and caught the adults on the same day. And with that, we also collected potential prey from all of the different territories. And then in 2011, we conducted nest feeding observations to try to validate our stable isotope model output and make sure that those two things were kind of saying the same message, so that we could infer from other years and other sites. So just to give a little bit of a background about stable isotopes, for those of you who don't really follow it a lot, basically the mantra is you are what you eat generally. So you have the same kind of isotopic signature of those things that you eat, and you can get basically that signature of what the consumer ate by different tissues. In this case, we're using blood, and you can separate plasma from the red blood cells to get 2 different time periods with which you can estimate what the consumer was eating. So plasma, generally you can determine what the organism was eating 10 to 24 hours before you caught it. And then with red blood cells is a little bit longer. It's about 10 days to a week. And so you can kind of use these to compare, especially for nestlings. The red blood cells would actually indicate what they were eating for their entire nestling period, whereas the plasma would indicate what nestlings are eating kind of when they're bigger, since we were catching them anywhere from 6 to 10 days in the nestling cycle. So the one problem with diet stable isotopes is you're not exactly what you eat, and there's a discrimination factor for whatever isotope that you're using, just because you assimilate at different rates. And so determining what this discrimination factor is kind of difficult. Generally, people assume carbon is 1 and nitrogen is 3. However, it's been found that those discrimination factors can actually vary quite a bit depending on tissue type, and in this case it's plasma and red blood cells, and then also for things that are growing quite quickly like a nestling would versus an adult. So we had to determine those discrimination factors for all of those different categories, and actually it was the most time-consuming part of the study was determining what those discrimination factors were. I'm not really going to get into that a whole lot though. I will just sort of give an example here is a graph showing carbon and nitrogen values for the sources that rusty blackbirds were eating potentially based on the nest feeding study. And you can see that they kind of discriminate nicely. They occur you know, in different areas of the graph, the different sources. There's some overlap, making it a little bit difficult to determine discrimination factors. But the black dots there are the original values of the tissue. In this case, it's red blood cells for rusty blackbirds. And the white dots are if you discriminate it by a carbon value of 1 and a nitrogen value of 3. So this is just an example. So what we did is we used mixing models to find the contributions of all of our diet sources to rusty blackbirds. And we did that for both the plasma and the red blood cells, like I said before, to kind of indicate short-term and long-term diet. And then we created movement variables from the telemetry data, and those movement variables were how far a bird moved from the nest in the first week, in the second week, and in the third week. And then we created a nest condition index that consisted of the number of nestlings and the coefficient of variation of the nest, and then adult condition index based on Peg and Green, which is a size-corrected body mass-based condition index. And then we used AIC model selection and program R to run different analyses for a couple of these questions. And we use site and season as random variables in all of those analyses. So briefly for the nest observations, we found that over a period of time from May 26th to June 16th in 2011, we had 12 nests and had 109 food observations, and this table is sort of a summary of all of the prey that we saw rusty blackbirds feeding to nestlings in 6 to 12-day-old nestlings. And what we found was, you see here on the right, this importance value is that dragonflies, adults, and larvae had the most importance, followed by caddisflies and then mayflies. So this was just the nest feeding observations, and this was just to validate what our isotopes models were telling us. This is the results from the isotope R modeling. And the main takeaway message from this is that the males, females, and hatch years had about the same composition of all of the different food items, which makes sense. I was sort of curious whether maybe they would be feeding nestlings something different than what they would eat themselves. Would they give nestlings larvae and they would take dragonfly adults for themselves? But it looks like pretty much that they're feeding the nestlings the same things they're eating themselves. And so on the bottom here, the x-axis, you can see that I've combined some categories. I have there's caddisflies, dragonfly adults, dragonfly nymphs, and then I combined dragonflies to see if that would be an interesting variable. And then I also combined larvae. And it ended up that the total larva was an interesting variable in further analyses. So this was the most interesting thing that I found about diet composition. So on the x-axis is the time from when the nests first hatched. So time 0 is the time that the very first nest we ever found hatched nestlings. And then on the y-axis, the proportion of larvae that those nestlings were modeled to have. And so what we're finding here seems to be that there's more larvae being incorporated into the diet as time goes on. Interesting or not, I'm not sure. This is the results of the movement analysis. And so what we found is there's a couple of top models that included the coefficient of variation of the nest. So what we found is that the most informative response variable was movement in a week, and CV nest, the amount of larva, total larva in red blood cells, and that was indicating the longer time frame, so over the course of the whole nestling period. And then the number of nestlings seemed to be describing how far they would move in a week. And all of these indicators were positive. However, when we actually determined the estimates, the confidence intervals were pretty large, and they also included 0, so that indicates that they were inconclusive, unfortunately. So the other thing that we wanted to look at was whether the body condition of adults was related to their food composition at all. And what we're seeing here is that it doesn't look like it, and there was no relationships with any of the other food sources, and males and females actually didn't really have any variation in their diet condition or in their size corrected body mass either. So it doesn't look like males are suffering from reduced condition than females, according to this index at least. So in conclusion, we found that there doesn't seem to be a relationship between food types and the condition of nestlings in adults. There doesn't seem to be a relationship between adult condition and nestling condition. Males and females had ate similar things, and actually, if you go back and look at that one graph, they're females and males from the same territories are basically eating exactly the same thing, according to our models. And then males and females have similar body condition, and the main conclusion that can be made from this study is that there was more larvae in the diet later in the season. So I think that just indicates that rusty blackbirds nest very early, and so maybe there's just not a whole lot of larvae early in the season, and more of those larvae are developing later on, and so they're able to incorporate more larvae in the diet. So this could have something to do with rusty blackbird timing of their nesting. And then the main conclusion, I guess, what we can get out of this is that the diet composition is likely not a limiting factor on the breeding ground, and that birds are able to find the correct composition of food that's required for them. All right, and this is the acknowledgments for this study. I'd like to thank the financial contributors, New Hampshire Audubon, Eastern Bird Branding Association, Smithsonian Migratory Bird Institution, the University of Georgia, and NSERC, which is a Canadian research institution, and then all of the field assistants that have helped out along the way, and some of the other supporters. The landowners are super amazing to work with, and other people who help collect insects and contribute blood samples. Thank you again for tuning in, and don't forget about our workshop. Thanks.